Hi, I am Maridely and I am going to present my poster titled Biosynthesis of Silver Nanoparticle with Pecan, Peanut, and Sunflower Seed for Colorimetric Sensing of Melamine. It is presented at the 18th Symposium of the 29th International Material Research Congress. Moving on to our abstract. Silver nanoparticles are of great interest for bioacademic and particle applications due to its plasmonic properties. Several applications have been reported for silver nanoparticles, such as catalysis of chemical reaction, nonlinear optics, fluorescence and colorimetric sensor, among others. Chemical synthesis of silver nanoparticles typically involves the use of harmful chemicals like sodium borohydrate or hydrazine hydrates and stabilizing agents like triazodium citrate or sodium lauryl sulfate. Also, depending on the desired application, something is necessary as surface functionalization of particles. Biosynthesis of silver nanoparticles by using plant extract is simple and economic. Moreover, an agroindustrial waste like seed and nut cell can be used as a raw material. In this work, we report the synthesis of silver nanoparticles by using pecan, peanut, and sunflower seed cell extract. The biosynthesis nanoparticle were tested for colorimetric zinc of melamine, and contrary to particles by peanut and sunflower seed thousand, particles synthesized pecan cell extract show a colorimetric response in presence of melamine. Obtaining the aqueous extract. The preparation of the extract was carried out by washing each of the cells with water. Subsequently, they were dried for 24 hours in the air. Once the drying times had elapsed, they were ground to increase the contact surface. Then different amounts were wet for its extract. To the peanut and sunflower extract were weighed to 100 milligrams, and for the pecan extract 50 milligrams were weighed. Later, 50 milliliters of distilled water were added and brought to a boil for 10 minutes. Finally, the extract was allowed to cool and be filtered. Finally, they were characterized by USB and FTAR. Synthesis of silver nanoparticles. The obtaining of silver nanoparticle was carried out by mixing 2 milliliters of a 25 millimolar silver nitrate solution with 10 milliliters of the pecan cell extract obtained in the previous step, and later it was measured at 50 milliliters. In the case of the peanut and sunflower cells, it was obtained by mixing 2 milliliters of a 25 millimolar silver nitrate solution with 20 milliliters of aqueous extract, and later it was measured at 50 milliliters. All experiments were allowed to reaction under ambient light for 45 minutes, subsequently stored in the dark for 2 days, and finally analysis by UV beams. Melamine detection. 3 milliliters of the silver nanoparticle dispersion were taken and a melamine solution was added in order to obtain a final concentration in the range of 100 millimolar to 400 millimolar. The mixture was allowed to reaction for 5 minutes and the USB spectrum was measured. The result obtained are polyphenols present in extract of dry fruit seed cell are a good reduction agent for the synthesis of silver nanoparticles. The extract of sunflower, peanut, and pecan nut cells were analyzed by USB. They present signal near to 176 nanometer that may correspond to an aromatic group. Sunflower and peanut present a signal near 340 nanometer that can be attributed to conjugate double bands, and pecan nut present a signal near 270 nanometer, which is attributed to the rising orange color in the solution. The nanoparticles synthesized with the shell extract were analyzed with USB, showing signal around 450 nanometer, corresponding to the signal of the silver nanoparticles. The nanoparticles were subject to melamine detection test using solution of 100, 200, and 400 millimolar of melamine. In this case of nanoparticles synthesized with sunflower and peanut seed cells, USB spectrum was not affected by the presence of melamine. Therefore, we can see that it's not possible to detect melamine of dozen concentration. However, in this case of the nanoparticles synthesized with pecan nut cell, the USB spectrum shows a slight shift and a shoulder appears between 500 and 600 nanometer when the melamine is added. The results confirm the qualitative detection of melamine by using silver nanoparticles synthesized with pecan nut cell. The conclusion. The green synthesis method here present by using peanut, sunflower, and pecan nut cell extract allow the synthesis of silver nanoparticles as can be confirmed by USB spectrum showing a signal at 450 nanometers. Besides a simple method to detect melamine in similar samples was tested. This method failed to detect melamine when peanut and sunflower seed cell extract are used for the synthesis of silver nanoparticles, but the detection of melamine is possible with silver nanoparticles synthesized with pecan nut cell.